the cosmic seed. Picture this, a fragment no larger than a grain of sand frozen for billions of years, travelling through the absolute darkness between stars. Inside that grain, wrapped in layers of ice and organic molecules, something waits, not conscious, not aware, but alive in the most fundamental sense. A bacterium, its cellular machinery suspended in a state so close to death that the difference becomes philosophical. And then, after a journey spanning light years and epochs, it crosses into our solar system aboard 3I Atlas, hurtling toward the inner planets at speeds that would vaporize anything less resilient. Before we dive deeper into what might be the most profound biological event in our planet's recent history, I want to ask you something. If you're finding value in these explorations of the unknown, hit that like button. It tells the algorithm that you want more of this content, more of these deep dives into the mysteries that keep us awake at night. And if you haven't already, subscribe and check for that new hype button some of you might see. Your support keeps these investigations going. Now, tell me in the comments, where are you listening from? What time is it there? And here's what I really want to know. If we discovered that life on Earth originally came from somewhere else, would that change how you see yourself and your place in the universe? Think about that while we continue. The theory we're exploring isn't science fiction. It's called panspermia, and it's been debated in scientific circles for over a century. The basic premise is elegant in its simplicity. Life, or at least its building blocks, didn't necessarily originate on Earth. Instead, it arrived here from somewhere else, carried by comets, asteroids, or in our case, an interstellar visitor that has already passed closer to Mars than most scientists ever dreamed possible. 3I Atlas isn't just any comet. When it swept past Mars at a distance of just 29 million kilometres, it was shedding material at a rate of 0.1 to 1 kilogram per second. Think about that for a moment. Every single second, between 100 grams and a full kilogram of this interstellar object's mass was being ejected into space. Dust particles ranging from microns to millimetres in size, each one potentially carrying the frozen remains of bacterial life that originated in a solar system we can't even identify. The mathematics of survival are staggering. Research on extremophile bacteria, particularly one nicknamed Conan the Bacterium, reveals something that sounds impossible until you see the data. This organism, officially called Deinococcus radiodurans, can withstand radiation doses 28,000 times greater than what would kill a human being. When frozen and dried, simulating the conditions it would experience in space or on Mars, it can survive for 280 million years if buried just 10 meters below the Martian surface. Even at a depth of just 10 centimeters, its lifespan extends to 1.5 million years. Now, let's connect these pieces. 3I Atlas is ancient. The cosmic ray bombardment it experienced during its journey altered its surface chemistry in ways that tell us it spent billions of years travelling between stars. If it carried microbial life when it left its home system, and if that life was anything like the extremophiles we've discovered on Earth, then those organisms could absolutely still be viable. Not just intact, but capable of reviving under the right conditions. The mechanism is more plausible than most people realise. Comets are not solid ice balls. They're complex structures with pockets and veins of different materials. In the coldest regions, far from the sun's heat, bacteria can remain in a state of suspended animation where molecular activity nearly stops. There's no metabolism, no reproduction, just the essential architecture of life preserved in crystalline ice. When 3I Atlas began its approach to the inner solar system, some regions heated up, but others remained frozen. The dust and debris it shed would have included both sterilized material from the heated zones and potentially viable organisms from the colder regions. Here's where the story takes a turn that should make everyone pause. When that material from 3I Atlas intersected with Mars's orbital path, the planet was right there. Not in terms of collision, we're not talking about an impact, but in terms of gravitational interaction and the simple geometry of where debris spreads in space. Mars doesn't have a thick atmosphere like Earth. Objects that would burn up completely before reaching Earth's surface can drift down onto Mars relatively intact, especially small particles in the micron to millimeter range. The research on this is fascinating and unsettling in equal measure. Scientists have tested how long bacteria can survive in simulated Martian conditions. They exposed multiple species to temperatures of -63 degrees Celsius, bathed them in ultraviolet radiation, subjected them to the low pressure of Mars' atmosphere, and bombarded them with the kind of cosmic radiation that constantly sleeting down onto the Martian surface. Several species survived, not just for hours or days, but long enough that the experiments had to be terminated with viable organisms still present. One study placed bacteria samples in pure water ice and in ice mixed with Martian regolith, the soil and rock material that covers the planet's surface. In pure ice, amino acids from E. coli bacteria remained intact for over 50 million years under Mars-like radiation conditions, even in ice mixed with Martian soil, where the degradation rate was faster, the survival time measured in the millions of years. The message is clear. If bacterial life made it to Mars' surface inside ice particles from 3I Atlas, it could still be there, dormant but not dormantly dead, waiting. But Mars is just the beginning of this story. 3I Atlas continued past Mars toward its closest approach to the Sun, then it curved back outward, and on its way out of the inner solar system, it will pass Earth. The distance is much greater, over 270 million kilometers, but the same principle applies. Material ejected from the comet spreads out in a vast cloud. Some of it, an infinitesimally small fraction, will intersect Earth's orbit. Most will burn up in our atmosphere, our planet's robust defense against space debris. But not all of it. Scientists studying meteor showers have known for decades that fine dust from comets reaches Earth's surface constantly. About 50 tons per day of extraterrestrial material drifts down through our atmosphere. The smallest particles, those in the micron range, don't heat up enough to sterilize their contents. They fall gently, taking weeks or months to complete their descent, protected by Earth's atmosphere from the worst of the cosmic radiation they encountered in space. Now, imagine that among those particles are fragments from 3I Atlas. Imagine that some of those fragments contain bacterial spores or cellular material from whatever form of life might exist in the comet's home system. What happens next? This is where evolutionary biology meets astrobiology in a way that's both thrilling and slightly terrifying. If alien bacteria reached Earth, they wouldn't arrive in a world ready to welcome them. They'd land in environments already occupied by terrestrial life that has had billions of years to optimize for every available niche. The competition would be fierce. Most alien organisms would fail immediately, unable to metabolize Earth's nutrients, overwhelmed by our oxygen-rich atmosphere if they evolved in an anoxic environment, or simply outcompeted by native species that are already perfectly adapted. But evolution doesn't require most to succeed. It only requires some. And here's where the research on horizontal gene transfer becomes critical. Bacteria don't just evolve through mutation and selection like larger organisms. They can exchange genetic material directly, swapping genes between species, even between domains of life that diverged billions of years ago. This process, called horizontal gene transfer, is arguably the most important mechanism in bacterial evolution. Studies have shown that bacteria can swap approximately 12% of their core genome in just 200 generations when they're in a proximity to other bacterial lineages. 200 generations might sound like a lot, but for bacteria that can reproduce every 20 minutes under ideal conditions, that's less than 3 days. Even under harsh conditions where reproduction takes hours or days, we're talking about timescales of months to years, not millennia. If bacteria from 3I Atlas made it to Earth, they wouldn't need to outcompete native life on their own. They would need to exchange genes with terrestrial bacteria. A novel metabolic pathway here, a radiation resistance gene there, a unique enzyme that allows breaking down a compound that Earth bacteria struggle with. Piece by piece, the alien and the terrestrial would blur together. The result wouldn't be an invasion in the science fiction sense. It would be a genetic merger, creating hybrid organisms that carry information from two different origins. Think about what this means. There could be bacteria living on Earth right now that carry genes from another solar system. Not ancient genes from the early solar system's formation, but recent acquisitions introduced within the last few months as 3I Atlas passed through our neighbourhood. These genes could be sitting silent in bacterial genomes, not expressed, not providing any obvious benefit, just waiting. Waiting for an environmental change that makes them valuable. Waiting for the right selective pressure to turn a neutral mutation into an adaptive advantage. The same process would be happening on Mars, but with crucial differences. Mars doesn't have the rich biosphere that Earth does. If life ever existed there, and the evidence keeps mounting that it did, it's either extinct or clinging to survival in isolated pockets. Subsurface ice, deep aquifers, anywhere liquid water might persist protected from the radiation and temperature extremes of the surface. In such an environment, bacteria from 3I Atlas would face less competition, but also harsher conditions. Here's the beautiful and strange part. Mars might be more welcoming to interstellar bacteria than Earth is, precisely because it's more hostile. On Earth, an alien bacterium has to compete with trillions of competitors in every gram of soil. On Mars, if it can survive the radiation and the cold and the desiccation, it might have entire niches to itself. The evolutionary pressure isn't to outcompete, it's simply to survive. And we know from Earth's extremophiles that life can survive in conditions we once thought were impossible. The research keeps revealing just how resilient microbial life can be. Bacteria have been found living in nuclear reactors, thriving in temperatures above boiling and below freezing, metabolizing rocks in the absence of any organic nutrients, and surviving years of exposure to hard vacuum and cosmic radiation. The experiments aren't theoretical. Scientists mounted bacterial cultures to the outside of the International Space Station and left them there for 3 years, directly exposed to everything space could throw at them. When they brought the samples back, some bacteria were still viable. Not just intact, but capable of reviving and reproducing once given water and nutrients. And now scale that resilience to the time scale of interstellar travel. 3I Atlas spent billions of years in the space between stars. The cosmic ray bombardment, the absolute cold, the complete absence of liquid water, none of it necessarily means biological death if the organisms involved are hardy enough and if they're protected inside ice and dust particles. The comet itself acts as a spaceship, shielding its interior from the worst of the radiation while maintaining temperatures low enough to prevent any molecular degradation. When 3I Atlas approached the Sun, regions of it heated up and began outgassing, releasing that material into space. The question isn't whether some bacteria might have survived the journey. The question is what happens next. What happens when those survivors encounter planetary environments? What happens when they meet native life forms, or when they find themselves alone on a world that once had life but lost it? This is the cosmic seed. Not a single organism, not even a single species, but a delivery mechanism. A way for genetic information to spread between stars not through radio signals or spacecraft, but through the ancient patient process of comets carrying their frozen cargo from solar system to solar system. If it happened once, it's happening constantly. Every interstellar object that passes through our neighbourhood is a potential vector. Every comet that the Sun captures temporarily from the Oort cloud might carry traces of biology from its origins around another star. The implications are staggering. It means that life on Earth might not have a single origin point. It means that the tree of life might have roots in multiple solar systems, braided together over billions of years through this process of cosmic exchange. It means that when we search for life on Mars, we might find organisms that share biochemistry with Earth life, not because they share a common ancestor on one planet, but because both planets have been recipients of the same interstellar seeding process. And here's the thought that should keep you awake tonight. If 3I Atlas brought life to Mars and Earth, where did that life come from originally? What conditions exist around other stars that might be pumping out these biological spores, sending them into the galaxy to find new homes? Is life rare, isolated to a few precious worlds, or is it common, constantly spreading, constantly adapting, using comets and asteroids as vehicles to colonize every suitable environment in the galaxy? The answers are buried in Martian ice, waiting for us to dig deep enough to find them. They're floating in Earth's stratosphere, settling onto our soil, every day unnoticed. They're encoded in the genomes of bacteria we haven't sequenced yet, silent passengers in the great experiment of terrestrial life. The cosmic seed has been planted. Now we need to discover what's growing from it. When 3I Atlas met Mars. The geometry was almost too perfect to be coincidence. On October the 3rd, 3I Atlas reached its closest approach to Mars, sweeping past at 29 million kilometres. In astronomical terms, that's practically a whisper away. The comet's trajectory brought it within the orbit of Mars at precisely the moment when the planet was positioned in just the right location. Some scientists calculated that with a velocity adjustment of merely 10 to 15 kilometres per second during September, the comet could have achieved a direct impact trajectory. It didn't. but the precision of the flyby raises questions that go beyond simple orbital mechanics. What matters for our story isn't whether the encounter was guided or random. What matters is what happened to all that material the comet was shedding. Remember, 3I Atlas was losing between 0.1 and 1 kilogram of mass every single second as it approached perihelion. That's not a gentle dusting. That's a constant spray of particles ranging from the microscopic to the size of small pebbles, each one following its own slightly different trajectory based on when and where it was ejected from the comet's surface. Mars Reconnaissance Orbiter, Mars Express, and the ExoMars Trace Gas Orbiter all turned their instruments toward the comet during the flyby. They captured images showing a coma several thousand kilometers across, though the exact boundaries were difficult to measure because the brightness fades gradually into the background noise. What they couldn't see, because the resolution simply wasn't there, were the individual particles, the dust grains in the micron to millimeter range that would be most likely to carry intact biological material. Here's what we know about how particles behave when a comet passes near a planet. They don't all continue along the comet's original path. Gravity, even Mars' relatively weak gravity, tugs at them. Solar radiation pressure pushes them. The particles spread out in a complex pattern, some accelerating ahead of the comet, some lagging behind, some deflecting toward the planet. The smallest particles, those in the micron range, are most affected by radiation pressure and can be blown into trajectories that intersect planetary orbits. Multiple spacecraft have been monitoring Mars for exactly this kind of event. The rovers on the surface, Perseverance and Curiosity, were both tasked with watching for any unusual atmospheric phenomena during the days surrounding the flyby. They didn't see anything dramatic, no visible meteor showers or sudden dust storms that could be attributed to the comet's debris, but that doesn't mean nothing reached the surface. Most of the particles small enough to carry protected bacteria would be too small to create visible meteors. They would simply drift down, taking days or weeks to settle through the thin Martian atmosphere. The critical question is whether any of those particles could have contained viable organisms. Let's walk through the sequence of events that a bacterium would experience if it survived this journey. First, it's frozen inside the comet's ice matrix for billions of years. Its cellular machinery is completely suspended, no metabolic activity whatsoever. As 3I Atlas approaches the Sun, parts of it of the comet heat up and begin sublimating, turning ice directly into gas. This process is violent. The escaping gas creates jets, carrying dust and larger fragments with it at the speeds that can exceed 100 meters per second. For most organisms, this would be instant death. The acceleration alone would shred cellular structures. But bacterial spores are different. They're built to survive exactly this kind of mechanical stress. Studies have shown that bacterial endospores can withstand accelerations of hundreds of thousands of times Earth's gravity. The pressures involved in cometary outgassing are well within their tolerance range. So step 1, ejection from the comet, is survivable for the right kind of organism. Step 2 is the transit through space from the comet to Mars. This is where radiation becomes the dominant threat. Cosmic rays are constantly bombarding everything in space, breaking chemical bonds and damaging DNA, but the particles aren't travelling alone through hard vacuum. Many are embedded in larger dust aggregates or ice crystals that provide some shielding. The journey from 3I Atlas to Mars would take anywhere from days to weeks depending on the particle's exact trajectory and velocity. During that time, radiation exposure would be significant but not necessarily lethal, especially for organisms with DNA repair mechanisms as robust as those found in extremophiles. Step 3 is entry into Mars' atmosphere. This is where Earth bacteria would face their greatest challenge, but Mars' atmosphere is only about 1% the density of Earth's. Entry heating is minimal for small particles. Something the size of a grain of sand would barely warm up at all. It would flutter down through the thin carbon dioxide atmosphere like a feather, taking its time, gradually settling toward the surface. No fireball, no plasma sheath, just a gentle descent. Step 4 is landing. Where a particle lands on Mars determines its fate. If it hits bare rock in the middle of the day when surface temperatures can briefly reach above freezing, ultraviolet radiation would sterilize it within hours. The Martian surface is bathed in UV light that would destroy any organic molecules exposed to it. But if the particle lands in shadow, if it settles into a crack or crater where dust can cover it, if it happens to come down in one of the polar regions where ice deposits accumulate, then survival becomes possible. The research on Martian ice is particularly relevant here. Scientists have mapped extensive ice deposits at both Martian poles and in mid-latitude regions. Some of these deposits are pure water ice, others are mixed with regolith. The studies on bacterial survival in different types of ice show dramatically different results. In pure water ice under Mars-like conditions, bacteria can survive for tens of millions of years. In ice mixed with Martian soil, survival times drop to hundreds of thousands of years due to chemical interactions between the ice, the soil minerals, and the radiation-damaged organic molecules. But here's what makes the 3I Atlas scenario so compelling. The particles settling onto Mars wouldn't have to survive for millions of years in their landing location. They'd need to survive long enough to reach a more protected environment. On Earth, we've discovered that bacteria can move through soil and rock layers, either by growing along moisture gradients or by being transported in flowing water. Mars doesn't have flowing water on its surface now, but it has ice. And where there's ice, especially in regions with geothermal heat, there's the possibility of liquid water in thin films between ice grains or in larger pockets deeper underground. Recent research has identified the most likely locations for liquid water on Mars. Subsurface lakes have been detected beneath the southern polar ice cap using ground-penetrating radar. These aren't lakes in the Earth sense, they were likely briny solutions with high concentrations of dissolved salts that keep them liquid at temperatures well below 0 degrees Celsius. But liquid is liquid. For bacteria, that's all that matters. The Perseverance rover has been examining rocks from an ancient river delta in Jezero crater. In September, the science team published findings on a rock formation they call Bright Angel. The rocks are fine-grained mudstones rich in oxidized iron, phosphorus, sulfur, and organic carbon. The patterns in these rocks show features that on Earth are only produced in specific ways. One way is through purely chemical processes at high temperatures. The other way is through biological activity, bacteria using chemical reactions between iron, sulfur, and organic matter as an energy source. The key detail is temperature. All the analyses of these Martian rocks indicate they were never heated above about 80 degrees Celsius. The chemical processes that could create these features without biology require much higher temperatures, several hundred degrees at minimum. If the rocks were never that hot, and they contain these specific patterns of minerals and organic compounds, then biology becomes the most plausible explanation. Not proof, but the most consistent hypothesis given the available data. Now connect this to 3I Atlas. If bacteria from the comet reached Mars and made their way into subsurface environments where liquid water exists, they'd encounter conditions similar to what generated those patterns in the Bright Angel rocks. Iron and sulfur are abundant in Martian regolith. Organic carbon might be sparse on the surface, but deeper down, protected from UV radiation, organic molecules from meteorites and from any native Martian biological activity could persist. The metabolic pathways that bacteria use to extract energy from iron and sulfur reactions are exactly the kind of universal biochemistry that could work anywhere in the solar system. There's another piece to this puzzle that's only recently come into focus. The Mars samples we've studied through rovers and from Martian meteorites that have fallen to Earth show evidence of microtunnels. These are tiny channels in the rock ranging from a few micrometers to tens of micrometers in diameter. They twist and curve through the mineral matrix in patterns that look remarkably similar to channels created by bacteria on Earth when they dissolve rock to extract nutrients. The morphology matches, the size matches, the distribution matches. What we can't prove yet is whether those tunnels were created by biological activity or by some purely chemical process we haven't identified. If bacteria from 3I Atlas established themselves in Martian subsurface environments, these microtunnels could be their signatures. The organisms wouldn't need to be alive today. They could have thrived for a few years, decades, centuries after arriving, then died out when conditions changed or resources were exhausted. But they would leave traces, microtunnels in rocks, unusual concentrations of specific isotopes, molecular fossils, fragments of cellular structures that resist degradation. The timeline is what makes this scenario different from previous speculation about Martian life. We're not talking about ancient Mars, billions of years ago when the planet had a thicker atmosphere and liquid water on the surface. We're talking about now. We're talking about an injection of biological material that happened months ago. If those organisms found a way to survive, they're alive right now, somewhere beneath the Martian surface, adapting to their new home. The adaptation process would be brutal and fast. Most organisms would die immediately. A few might survive in a dormant state waiting for better conditions. An even smaller number might find themselves in an environment where they can actually metabolize, grow and reproduce. Those are the ones that matter for evolution. Those are then the ones that would begin the process of becoming Martian. The selective pressures would be intense. Water availability would be the first constraint. Any bacterium that couldn't extract water from ice or tolerate extremely high salt concentrations would die. Radiation resistance would be second. Even underground, cosmic rays and radioactive decay from Martian rocks create a constant background of ionizing radiation. Energy sources would be third. There's no sunlight underground. No organic matter raining down from a biosphere above. The only energy available comes from chemical reactions between minerals and the extremely limited dissolved gases in whatever liquid water exists. But bacteria are survivors. They've colonized every environment on Earth, from kilometers deep in the continental crust to the upper atmosphere. They've adapted to conditions we keep thinking should be impossible. Boiling acid, pure salt, solid rock, hard vacuum. If bacteria from 3I Atlas carried the right genetic toolkit, if they had genes for radiation resistance, for extracting water from minerals, for metabolizing simple chemical reactions, then Mars could become their home. And here's the truly fascinating possibility. If bacteria from 3I Atlas did successfully colonize Martian subsurface environments, we might never detect them with current technology. Our rovers can only drill a few meters down. The ExoMars rover, which was supposed to drill deeper, has been indefinitely postponed. The ice deposits where life is most likely to survive are buried beneath meters to kilometers of rock and soil. We'd need to mount a dedicated mission specifically designed to reach those depths, extract samples, and analyze them for biological signatures. Which means that right now, as we orbit Mars with our spacecraft and roll across its surface with our rovers, there could be an entire biosphere developing beneath our sensors. Not native Martian life, not ancient fossils, but a new ecosystem born from an interstellar delivery just months ago. The bacteria would be dividing, mutating, adapting. In a few generations, they wouldn't look anything like their ancestors from 3I Atlas's home system. They'd be Martian bacteria, optimized for Martian conditions, carrying genetic information from another star but shaped by the red planet's unique environment. The encounter between 3I Atlas and Mars wasn't just an astronomical event. It was potentially a biological one, a moment when two separate lineages of life's history intersected. What grows from that intersection is still unfolding, hidden from view, waiting to be discovered. The survival equation. Let's talk about what it actually takes to survive in space. Not the science fiction version with shielded spacecraft and life support systems, but the raw, unprotected reality of hard vacuum, cosmic radiation, and temperatures that swing from barely above absolute zero to scorching heat depending on whether you're in shadow or sunlight. Because if we're going to seriously consider bacteria travelling aboard 3I Atlas, we need to understand the actual physics and biology of survival at the extremes. Conan the bacterium earned its nickname honestly. When researchers exposed Deinococcus radiodurans to radiation doses that would kill a human 28,000 times over, the organism didn't just survive, it thrived. After receiving gamma-ray doses totaling 140,000 Gray, about 28 times what would completely sterilize medical equipment, the bacteria repaired their shattered DNA and continued reproducing. The mechanism is elegant. These organisms maintain multiple copies of their entire genome, and they possess extraordinarily efficient DNA repair enzymes that can essentially rebuild the genetic code from fragments. But radiation resistance is just one piece of the survival equation. Temperature poses an equally serious challenge. In the shadow of an interstellar object travelling through space, temperatures hover around minus 270 degrees Celsius, just 3 degrees above absolute zero. At these temperatures, all chemical reactions essentially stop. Water ice becomes harder than steel. Proteins can't fold or unfold, nothing moves. For most organisms, this is death. For bacterial spores, it's preservation. The spore form is one of evolution's most brilliant solutions to hostile environments. When conditions deteriorate, certain bacteria don't just slow down their metabolism, they dismantle it. The spore surrounds its DNA with protective proteins and encases everything in multiple layers of specialized membranes and a thick coat. Then it purges almost all its water content, creating a desiccated capsule where biochemistry can't happen because there's no liquid medium for reactions to occur in. In this state, the organism isn't alive in any meaningful sense. It's not dead either, it's waiting. International Space Station experiments prove the concept works in actual space conditions. Researchers mounted bacterial cultures on the exterior of the station, directly exposed to vacuum, radiation, temperature extremes and solar UV. After 3 years, they brought the samples back. Some bacteria were still viable, not just chemically intact, but capable of reviving when given water and nutrients. The survival rate wasn't high, maybe 1 cell in 10 million, but that's the point. Evolution doesn't require high survival rates, it requires any survivors at all. The mathematics of probability start to work in favour of life when you consider the sheer scale involved. 3I Atlas has a diameter of at least 5 kilometres. That's a volume of approximately 65 cubic kilometres if we assume a roughly spherical shape. If even a tiny fraction of that volume contained bacterial spores at the kind of concentrations found in Earth's soil, we're talking about billions of trillions of individual organisms. When the comet shed material near Mars, even if only one particle in a billion contained viable bacteria, and even if only one bacterium in a million from those particles survived the journey and landing, you'd still have thousands of organisms reaching the Martian surface alive. But survival isn't just about the journey, it's about what happens after arrival. This is where the specific conditions on Mars become critical. The planet's surface is hostile, but it's not uniformly hostile. Radiation levels vary dramatically depending on location and depth. At the surface in equatorial regions, the radiation dose is about 76 milligray per year, roughly equivalent to getting 40 full-body CT scans annually. That's enough to prevent most life as we know it from surviving long-term, but dig down just 2 meters, and you're behind enough shielding to drop the radiation exposure by 90% or more. The temperature profile on Mars is equally variable. Surface temperatures can briefly exceed 0 degrees Celsius at the equator during summer afternoons, but they plunge to minus 125 degrees at night. The subsurface is different. A few meters down, the temperature stabilizes, insulated from the daily and seasonal swings. In regions with geothermal activity, even minimal heat flow from the planet's interior can keep subsurface ice deposits at temperatures where liquid water can exist if the right salts are dissolved in it. Perchlorate salts are abundant in Martian soil, and they're both a threat and an opportunity for bacterial life. In high concentrations, perchlorates are toxic, oxidizing cellular structures and disrupting metabolism. But some Earth bacteria have evolved to not only tolerate perchlorates but use them. Certain species can metabolize perchlorate as an energy source, breaking it down and using the released oxygen for respiration even in environments completely devoid of gaseous oxygen. If bacteria from 3I Atlas carried similar capabilities, or if they could acquire those genes through horizontal transfer from any native Martian organisms, perchlorate-rich environments would transform from barriers to habitats. The energy budget for subsurface bacterial life on Mars would be tight but manageable. On Earth, bacteria living kilometers deep in rock extract energy from the oxidation of hydrogen gas, produced by chemical reactions between water and iron minerals. The process is slow. These bacteria can take centuries to reproduce once, making them the slowest growing organisms known. But slow growth is still growth. It's still evolution. It's still life continuing. Mars has plenty of iron minerals. The planet's red colour comes from iron oxide, rust, covering nearly every surface. Deeper down, unoxidized iron reacts with any available water to produce hydrogen gas. That hydrogen becomes fuel. Bacteria that can capture it and use it to reduce carbon dioxide into organic molecules have an energy source that doesn't depend on sunlight or organic nutrients from above. They become primary producers, the bottom of a food chain that has exactly one member, feeding only on chemistry. Laboratory simulations of Martian subsurface conditions have produced surprising results. When researchers create environments matching Mars's temperature, pressure, mineral composition, and radiation levels then introduce bacteria, some species don't just survive, they adapt. Over hundreds of generations in these harsh conditions, the bacteria evolve. Mutations that would be neutral or even slightly harmful in Earth conditions become beneficial. Genes that were rarely expressed suddenly become critical. The organisms don't become less complex, they become differently complex, optimized for a completely new set of constraints. One particularly revealing study examined how bacterial metabolism changes under Martian pressure and temperature. At 7 millibars of pressure, about 0.5% of Earth's sea level, pressure, and at temperatures just above freezing, only one bacterial species out of 26 tested showed growth. That species, Serechia liquefactions, managed to reproduce despite conditions that stopped all its relatives. The key was its ability to maintain cellular pressure gradients, even when the external pressure dropped below what most organisms can tolerate. A single metabolic trick, probably controlled by just a handful of genes, made the difference between death and survival. This is why horizontal gene transfer becomes so crucial in our story. If bacteria from 3I Atlas landed on Mars carrying completely alien biochemistry, they'd almost certainly die. The mismatch between their genetic programming and the Martian conditions would be too great. But if those organisms carried genes that are functionally similar to what Earth bacteria use, just tweaked and optimized for different conditions, then gene exchange becomes possible. A Martian bacterium, if any exist, could pick up radiation resistance genes from the newcomers. An alien bacterium could acquire pressure tolerance genes from a native Martian survivor. Both lineages benefit. Both increase their chances of survival. The mechanism for gene transfer doesn't require direct cell contact in the way it often does on Earth. In extremely low nutrient environments, bacteria release DNA fragments when they die. Those fragments can persist in the environment, especially in ice or in mineral pores, for thousands of years. When a living cell encounters those fragments, it can sometimes incorporate them directly into its own genome. The process is called transformation, and it's one of the ways antibiotic resistance spreads so quickly in bacterial populations. What works in a hospital can work in Martian ice. There's another factor that makes Mars potentially more hospitable than it first appears. The planet's low gravity, about 38% of Earth's, means bacterial cells experience less physical stress. On Earth, bacteria in deep subsoil have to cope with enormous pressures from the overlying rock. On Mars, the same depth results in much lower pressure. Cellular structures don't have to be as robust. Resources don't have to be diverted to maintaining structural integrity. That's energy that can be used for growth and reproduction instead. The water situation on Mars is more complex than the simple statement that liquid water can't exist on the surface. That's true for pure water, but salt solutions behave differently. Perchlorate brines can remain liquid at temperatures as low as -70 degrees Celsius. At those temperatures, metabolism would be incredibly slow, but not zero. Enzymes would still function, just at rates thousands of times slower than in warm water. For organisms with lifespans measured in millennia, that's acceptable. Recent discoveries have identified locations on Mars where conditions seem optimized for microbial survival. The recurrent slope lineae, dark streaks that appear seasonally on some Martian slopes, might represent brine flows. Radar detection of subsurface liquid water beneath the South Polar ice cap suggests entire lakes of salty water existing in the dark. Gullies and crater walls show evidence of recent fluid flow, possibly from the eruption of subsurface aquifers. Each of these environments could harbour life if organisms could reach them and adapt. The survival equation ultimately comes down to a few key variables. First, radiation shielding, solved by being underground. Second, temperature management, solved by finding geothermally influenced regions or by simply accepting extremely slow metabolism in frozen conditions. Third, water access, solved by utilising brines that remain liquid below freezing. Fourth, energy sources, solved by chemosynthesis using minerals and simple gases. Fifth, genetic flexibility, solved by horizontal gene transfer, allowing rapid adaptation. Every one of these variables has solutions demonstrated by Earth organisms. Every one of these solutions involves biochemistry that would work on Mars with little or no modification. The question isn't whether bacterial life could survive on Mars. Earth bacteria have already proven they can in laboratory simulations. The question is whether bacteria from 3I Atlas brought the right genetic toolkit with them, and whether they landed in locations where survival was possible. If they did, then somewhere beneath the Martian surface, the survival equation is being solved right now. Cells are dividing, DNA is mutating, selection is operating. The great experiment of life adapting to a new world is underway, and we're only just beginning to suspect it might be happening. First contact with Martian soil, the moment a bacterial spore from 3I Atlas touches Martian regolith. A cascade of chemical reactions begins. Not biological reactions, not yet, but the purely physical and chemical processes that determine whether revival is even possible. The spore, perhaps embedded in a tiny ice crystal or dust aggregate, settles onto oxidized iron minerals. The landing site might be a crater floor, a valley system, or perhaps one of the vast dusty plains that cover much of Mars' northern hemisphere. Martian soil is nasty stuff from a biological perspective. It's loaded with oxidizing compounds, particularly those perchlorate salts we discussed, but also hydrogen peroxide and other reactive oxygen species. When the Viking landers ran their biology experiments in the 1970s, they got results that initially looked like life. The soil samples produced gas when given nutrients and water. But further analysis revealed something more troubling. The Martian soil itself was so chemically reactive that it destroyed organic molecules on contact. What looked like biological activity was actually violent chemistry. This reactivity comes from Mars's lack of a magnetic field and thick atmosphere. On Earth, our magnetosphere deflects most cosmic radiation. Our ozone layer absorbs the harshest ultraviolet light. Mars has neither. Solar radiation and cosmic rays hit the surface unimpeded, breaking chemical bonds in soil minerals and creating these reactive compounds. Over billions of years, the entire surface has been processed into a sterilizing environment. But here's the critical detail that changes everything. That sterilization only penetrates to a certain depth. UV radiation can't pass through raw rock or even through layers of dust. Cosmic rays are more penetrating, but their effects decrease dramatically with depth. A bacterial spore buried under just a few centimeters of soil is protected from the worst of it. If the spore can survive the initial contact with the surface material and get buried, either by settling dust, wind action, or by landing in a crack or crevice already protected from direct radiation, it enters a completely different environment. Subsurface Martian soil still contains oxidizing compounds, but at much lower concentrations. The minerals are less processed, more like the original rocks that formed them. There might even be organic molecules preserved from ancient impacts of carbonaceous asteroids, molecules that couldn't survive at the surface but remain intact meters below. This is the environment where a bacterium from 3I Atlas would have its first real chance of survival. The spore needs water, not much, just enough to trigger the revival process. In a spore's dormant state, water content is below 5%. The cellular machinery is disassembled and stored. DNA is protected by specialized proteins that prevent damage from radiation and chemical attack. To revive, the spore must rehydrate, taking in water molecules that will allow enzymes to refold and begin functioning. On Mars, that water is scarce, but it's not absent. Thin films of water can exist on mineral surfaces even in conditions that seem completely dry. When humidity is present, even at Martian pressures, water molecules adhere to mineral grains through chemical bonding. These films are only a few molecules thick, but for a bacterium it might be enough. The spore begins to absorb water from its surroundings gradually over days or weeks. As water content increases, the protective proteins around the DNA start to release their hold. The genetic code becomes accessible again. Now comes the first critical test. Is the DNA intact? Cosmic radiation during the journey from 3I Atlas will have caused damage. Single-strand breaks are almost certain. Double-strand breaks are likely. Some genes might be completely destroyed, their information lost. But bacteria carry redundancy. Many extremophiles maintain multiple copies of their entire genome, specifically as insurance against radiation damage. If even one copy remains mostly intact, the repair enzymes can begin reconstruction. The repair process is remarkably sophisticated. Enzymes scan along the DNA, identifying damaged sections. They cut out broken pieces and use the information from undamaged copies to fill in the gaps. It's not perfect. Some errors creep in, some genes are modified in the repair process, but those modifications are mutations, the raw material of evolution. If the mutations happen to be beneficial in Martian conditions, they'll be preserved when the cell divides. Assuming the DNA repairs successfully, the bacterium faces its next challenge. It needs energy. The stored energy reserves in a spore are minimal, just enough to power the revival process and perhaps a few rounds of basic metabolism. After that, the organism must find an external energy source or die. This is where biochemical compatibility with the Martian environment becomes critical. If the bacterium evolved around a star similar to our Sun, on a planet with liquid water and a reducing atmosphere, its metabolic pathways might be very similar to Earth bacteria. Carbon dioxide as a carbon source, see hydrogen as an electron donor. Simple sulfur or iron compounds as electron acceptors. These are universal chemical reactions that work anywhere liquid water exists. Mars has carbon dioxide in abundance, it's the primary atmospheric gas, and it's dissolved in any liquid water that exists. Mars has hydrogen from water-rock interactions. Mars has sulfur and iron everywhere. The bacterium extends molecular machinery through its outer membrane, testing its environment. Chemical sensors probe for available nutrients. If it finds what it needs, metabolism begins. Slowly at first, because the temperature is likely well below freezing even underground. But chemistry still happens in supercooled brines. Enzymes still function, just at dramatically reduced rates. The bacterium might take weeks to accumulate enough energy for a single cell division. That's acceptable. Time is unlimited underground. But there's competition for resources, even in the Martian subsurface, if any native Martian bacteria exist. The newcomer from 3i Atlas would encounter organisms that have had billions of years to optimize for this environment. They would be better adapted, more efficient, faster at scavenging the limited nutrients. The alien bacterium would be at a severe disadvantage, unless it brought something new. This is where the genetic cargo from another star system becomes valuable. Perhaps the alien carries genes for metabolizing a compound that Martian bacteria never evolved to use. Perhaps it has more efficient enzymes for capturing hydrogen at extremely low concentrations. Perhaps it tolerates higher salt levels or lower temperatures. Any edge, any novel capability creates a niche where the newcomer can survive alongside the natives rather than being outcompeted. Gene transfer would begin almost immediately. Dead bacterial cells release their DNA into the environment. Both the aliens and the natives would encounter these fragments. A Martian bacterium might pick up a gene cluster from the newcomer. An alien might acquire a cold tolerance gene from a native. Over hundreds of generations, the distinction between native and alien would blur. Hybrid genomes would emerge, carrying the best adaptations from both lineages. There's evidence this process happens rapidly when bacteria colonize new environments. Studies of bacterial populations in extreme Earth environments show that when multiple species encounter each other, gene transfer accelerates dramatically compared to laboratory conditions. The selective pressure to acquire useful genes is intense. Organisms that happen to pick up beneficial DNA fragments outcompete their relatives. The process snowballs. Within a few thousand generations, you can see dramatic changes in population genetics. The physical structure of Martian subsurface environments would influence where and how bacterial communities develop. Pore spaces in rocks, ranging from nanometers to millimeters in size, provide protected microhabitats. Fractures and faults create pathways for fluid movement. Ice lenses and layers offer both water sources and physical structure. Each of these environments selects for different bacterial traits. In fine-grained sediments with tiny pore spaces, only the smallest bacteria survive. Size becomes a critical factor. In larger fractures with more space, biofilms can develop. Communities of bacteria embedded in self-produced matrix materials. Biofilms offer advantages. They create local chemical gradients that concentrate nutrients. They provide some protection from environmental stress. They facilitate gene transfer between adjacent cells. If bacteria from 3i Atlas established biofilm communities in Martian fractures, those communities could persist for extraordinarily long times. The key resources that would limit bacterial growth in Martian subsurface environments are water, phosphorus, and nitrogen. Water availability we've discussed. It exists but requires accessing brines or thin films on mineral surfaces. Phosphorus is essential for DNA, RNA, and ATP, the energy currency of cells. Mars has phosphorus in minerals, but extracting it requires dissolving those minerals, which requires liquid water and time. Nitrogen is needed for proteins and nucleic acids. Mars's atmosphere is mostly carbon dioxide with only trace nitrogen. Subsurface bacteria would need to scavenge nitrogen from minerals or from any dissolved nitrate or ammonium in groundwater. These limitations mean Martian bacterial populations, whether native or introduced, would grow slowly and remain at low densities. There wouldn't be the thick mats of bacteria you see in Earth's soil. Instead, you'd have sparse populations, perhaps a few million cells per gram of rock rather than the billions per gram typical of Earth. But that's still life. That's still ecology. That's still evolution operating. The really intriguing possibility is that bacteria from 3I Atlas might find Martian subsurface environments actually easier to colonize than Earth. On Earth, every habitat is already full of competitors. Newcomers have to fight for every resource. On Mars, if native life is extinct or restricted to isolated pockets, there are ecological niches sitting empty. A bacterium that can tolerate the conditions doesn't have to outcompete anyone. It just has to survive and reproduce. The absence of competition makes up for the harshness of the environment. Over time, assuming survival and reproduction, the population would diversify. Different lineages would adapt to different microhabitats within the subsurface. Some would specialize in extracting phosphorus from specific minerals. Others would optimize for capturing trace hydrogen. Still others would develop tolerance for even higher perchloric concentrations, allowing them to exploit resources other bacteria couldn't access. The single founding population would radiate into multiple ecological roles. This is adaptive radiation, the same process that produced Darwin's finches on the Galapagos Islands. Start with one colonizing species in an environment with available niches. Let evolution operate for enough generations. The result is diversification, specialization, and the emergence of entirely new species adapted to specific conditions. On Mars, the timescale would be longer because bacterial reproduction is so slow in the cold. But the principle is the same. The first contact between bacteria from 3I Atlas and Martian soil isn't a single event. It's an ongoing process that started when the first particles settled months ago and will continue for as long as viable organisms exist. Every spore that revives is another trial, another test of whether life from elsewhere can make a home on Mars. Most fail, some might succeed, and those successes, invisible to our current senses, could be writing a new chapter in the biological history of two worlds. The horizontal revolution evolution, by vertical inheritance, parent to offspring, generation after generation, is the story we learned in school. But it's not the only story. For bacteria, and possibly for life throughout the universe, horizontal gene transfer is equally important and far more dramatic. It's the difference between waiting millions of years for beneficial mutations to arise randomly and acquiring those same beneficial traits in a single generation by copying them from a neighbour. The numbers are staggering. Research has shown that bacteria can exchange approximately 12% of their core genome in just 200 generations when two compatible lineages encounter each other. 200 generations sounds like a long time for humans, but for bacteria reproducing even once per week in harsh conditions, that's less than 4 years. In more favourable environments where division occurs daily, we're talking about months. This isn't gradual accumulation of tiny changes, this is wholesale swapping of genetic modules, entire pathways and capabilities moving between organisms as if bacteria were sharing software code. There are 3 primary mechanisms for horizontal gene transfer in bacteria. Conjugation involves direct physical contact between cells, with one bacterium extending a structure called a pilus to another and pumping DNA through it. The transferred DNA is typically carried on plasmids. Small circular DNA molecules separate from the main chromosome. These plasmids often carry genes for antibiotic resistance, metabolic capabilities, or environmental tolerance. When a bacterium acquires a plasmid, it gains all those capabilities instantly. Transformation is simpler but requires the right conditions. Bacteria can take up naked DNA from their environment, DNA released when other cells die and break apart. This might seem like a random process, but some bacteria actively promote it. They develop competence, a physiological state where their membranes become permeable to large molecules like DNA. They'll grab fragments from the surrounding medium and incorporate them into their own genome through recombination. If those fragments happen to contain useful genes, those genes become part of the cell's permanent genetic repertoire. Transduction involves viruses as vectors. Bacteriophages, viruses that infect bacteria, sometimes accidentally package host DNA along with their own genetic material. When they infect the next bacterial cell, they inject that DNA. If the injected DNA contains genes that help the new host survive, natural selection will favour bacteria that retain those genes. Phage-mediated gene transfer is common in aquatic environments on Earth. Whether phages exist on Mars or arrived with bacteria from 3I Atlas is unknown, but if they do, they'd accelerate the mixing of genetic information between any existing bacterial populations. What makes horizontal gene transfer revolutionary for adaptation is the way it navigates fitness landscapes. In traditional Darwinian evolution, a population climbs gradually up a fitness peak through accumulated mutations. But what if there's a higher peak separated by a valley of low fitness? The population would have to get worse before it could get better, and natural selection actively prevents that. Horizontal gene transfer solves this problem by allowing organisms to jump directly from one peak to another without crossing the valley. A bacterium struggling in one niche can acquire genes that make it competitive in a completely different niche, all in one step. Laboratory experiments have demonstrated this dramatically. Researchers evolve bacterial populations under controlled conditions, then introduce DNA from bacteria adapted to different environments. Populations that have access to foreign DNA adapt faster than populations relying solely on mutation. They explore more of the possible genetic space. They find solutions that mutation alone would take thousands of generations to discover. The effect is, most pronounced when the environment is harsh or rapidly changing, exactly the conditions bacteria from 3I Atlas would encounter on Mars. One particularly revealing study examined bacterial adaptation to antibiotic exposure. Populations were grown in environments without antibiotics but with access to DNA from antibiotic-resistant strains. Over time, the non-resistant bacteria picked up resistance genes at low frequency. These genes didn't provide any benefit in the antibiotic-free environment, so they remained rare in the population. But when antibiotics were suddenly introduced, populations with those rare resistance genes exploded in number, while populations without access to foreign DNA went extinct. Horizontal gene transfer didn't just accelerate adaptation, it enabled survival in conditions that would otherwise be lethal. This has profound implications for bacteria from 3I Atlas encountering Martian conditions. Even if the alien organisms are poorly adapted initially, even if they struggle to metabolize Martian chemistry or tolerate Martian temperatures, they can acquire the necessary genes from native Martian bacteria if any exist. Conversely, native Martian organisms could acquire novel capabilities from the newcomers. Both populations benefit, both become more resilient. The rate of horizontal gene transfer depends on several factors. Physical proximity is crucial. Bacteria need to be close enough for conjugation to occur, or for released DNA to reach competent cells before it degrades. Environmental DNA doesn't last forever. Enzymes called nucleases, present even in the Martian environment from degraded cells, break down exposed DNA over time. In Earth soils, naked DNA typically persists for days to weeks. In Martian ice or frozen brine, it could last much longer, perhaps years or centuries, because the cold inhibits enzymatic activity. Genetic compatibility is another factor. Not all DNA can be successfully integrated into any genome. There are molecular mechanisms that protect bacteria from incorporating foreign DNA that's too different. Restriction modification systems, essentially bacterial immune systems, cut up foreign DNA that lacks the right molecular markers. But these systems can be overcome. If the transferred DNA provides a strong enough survival advantage, bacteria that fail to restrict it will outcompete bacteria that do. Over time, the population becomes more permissive to gene transfer. The structure of bacterial genomes facilitates this kind of genetic mixing. Unlike eukaryotic organisms with tightly organized chromosomes, bacteria often have loosely arranged genomes with frequent insertion sequences and mobile genetic elements. These are stretches of DNA that can move around within the genome or jump between different DNA molecules. They create natural recombination hotspots where foreign DNA can insert. Once inserted, if the genes are beneficial, they're retained. If they're harmful, they're lost, through deletion or by being outcompeted by cells that didn't acquire them. One of the most powerful aspects of horizontal gene transfer is that it can move entire functional modules at once. On Earth, genes for related functions often cluster together in operons, groups of genes transcribed as a single unit. When an operon transfers between bacteria, the recipient gains a complete metabolic pathway or regulatory system all at once, not piece by piece. This is why antibiotic resistance can spread so quickly. A single plasmid might carry resistance genes for 5 different antibiotics plus the regulatory elements that control their expression. One transfer event creates multi-drug resistance. If bacteria from 3I Atlas carry genes organized in operons, those complete functional units could transfer to Martian bacteria as intact modules. A cluster of genes for radiation resistance, a pathway for metabolizing perchlorate, a system for extracting phosphorus from specific mineral types. Each transfer would be a major adaptive leap, not an incremental improvement. This is evolution operating in fast-forward, species-level change occurring in the time it would normally take for a single point mutation to spread through a population. There's also a fascinating feedback loop. Horizontal gene transfer becomes more common in stressful environments because stress induces competence. When bacteria are starving or damaged, they're more likely to take up foreign DNA. It's a desperate measure, a last roll of the dice before death. Most of the DNA they acquire is useless or harmful, but occasionally, purely by chance, they grab genes that solve their problem. Those rare successes are what natural selection preserves and amplifies. Mars is the definition of a stressful environment for bacteria: low nutrients, extreme cold, radiation, chemical toxicity. Every bacterium, whether native or introduced, would be under constant stress. That stress would drive high rates of competence and transformation. DNA release from dying cells would be continuous. Surviving cells would be constantly sampling that environmental demi pool. The rate of genetic exchange could be orders of magnitude higher than in the comfortable environments where most laboratory studies are conducted. There's another wrinkle that makes this scenario even more plausible. Gene transfer networks on Earth show strong ecological structure. Bacteria in the same habitat exchange genes more frequently than bacteria in different habitats, even if they're closely related species. The explanation is simple. Genes that help in one environment might be useless or harmful in another. Bacteria benefit most from acquiring genes from neighbours dealing with similar challenges. This means that even if bacteria from 3I Atlas are genetically distant from any native Martian life, if they end up in similar habitats facing similar problems, gene transfer between them becomes likely. Recent research has revealed that in natural bacterial communities, a significant fraction of successful gene transfers involve genes related to surface structures, nutrient transport, and stress response. These are exactly the kinds of genes that would be most valuable in Martian subsurface environments. Genes for modified membrane structures that function in high salt or low temperature. Genes for transporters that scavenge trace nutrients. Genes for repairing radiation damage or tolerating oxidative stress. These functional categories would be under intense selection pressure on Mars. The long-term result of high rates of horizontal gene transfer is that bacterial populations become genetic mosaics. Instead of clear phylogenetic trees showing ancestry, you get networks showing complex patterns of gene sharing. An organism's genome becomes a patchwork. Some genes inherited vertically from its ancestor, others acquired horizontally from distant relatives, still others picked up from completely unrelated lineages. The concept of species becomes fuzzy for bacteria because genetic boundaries are so permeable. On Mars, this could mean that within a few thousand generations, you couldn't distinguish bacteria descended from 3I Atlas from bacteria descended from native Martian life, if any exists. Both lineages would have exchanged so many genes that their genomes would be hybrids. They'd all be Martian bacteria, uniquely adapted to their planet, carrying genetic information from multiple origins blended into new combinations that never existed anywhere else in the universe. The horizontal revolution isn't just about faster adaptation. It's about expanding the realm of the possible. It allows organisms to access genetic solutions that mutation alone might never discover. It enables rapid response to environmental change. It creates biological diversity without waiting for speciation. And it means that life, once introduced to a new world, doesn't have to reinvent every adaptation from scratch. It can borrow, steal, and remix the genetic innovations of whatever other life it encounters, building something new from the combined wisdom of separate evolutionary histories. Earth's uninvited guests. Their Atlas passed Mars at 29 million kilometers. Its closest approach to Earth will be 270 million kilometers, nearly 10 times more distant. But distance isn't the only factor that matters when we're talking about microbial dispersal. What matters is the debris cloud, the vast expanding sphere of particles that the comet shed as it approached perihelion and continues to shed as it moves away. That cloud is big enough and diffuse enough that some fraction of it will intersect Earth's orbit. The math is humbling. 3I Atlas was ejecting between 0.1 and 1 kilogram of material every second for months. Let's be conservative and say an average of half a kilogram per second sustained over 100 days around perihelion. That's over 4 million kilograms of dust and ice particles released into orbits that now crisscross the inner solar system. Most of that material will never come near Earth. It'll drift outward with the comet or fall inward toward the Sun, but statistical mechanics tells us that some fraction, however small, will end up on trajectories that bring them to our planet. Earth already intercepts about 50 metric tons of extraterrestrial material every day. Most of it is dust from comets and asteroids, particles so small they drift down through the atmosphere without burning up. They settle onto soil and ocean surfaces, adding a constant sprinkle of cosmic material to our planet's surface layer. We've found these particles in ice cores in deep sea sediments in the stratosphere. They are everywhere, and mostly we ignore them because they're inert. Just minerals and occasional organic molecules, the leftover construction material from the solar system's formation. But what if some of the particles settling onto Earth over the next few months aren't ancient debris? What if they're fresh ejecta from 3I Atlas, carrying biological material from another star system? The implications are profound and deeply unsettling. Earth isn't like Mars. We have a thick atmosphere, a strong magnetic field, abundant water, and most critically, biosphere that has spent 4 billion years filling every available ecological niche. Newcomers don't get a warm welcome. The first barrier is atmospheric entry, and even particles in the micron range experience heating as they collide with air molecules during their descent. For larger particles, millimeters in diameter, the heating can be extreme. The outside surface chars and ablates away, but the interior, protected by the outer layers, can remain relatively cool. Studies of micrometeorites recovered from Earth's surface show that, while the outsides are melted and oxidized, the cores often contain pristine minerals and even organic compounds that never reach destructive temperatures. If a particle from 3I Atlas containing bacterial spores enters Earth's atmosphere, the spores on the surface will be sterilized instantly. But spores embedded deeper inside, particularly in ice-cemented dust aggregates, might survive. The ice would ablate away during entry, providing cooling through sublimation. By the time the particle slows to terminal velocity in the lower atmosphere, it would be cold again. Any surviving spores would arrive on Earth's surface viable but dormant. Where they land determines what happens next. If they land in the ocean, they're entering the most biologically competitive environment on the planet. Marine bacteria are extraordinarily diverse and aggressive. They produce antimicrobial compounds. They form biofilms that exclude newcomers. They've evolved for billions of years to defend their territories. An alien bacterium, even one with sophisticated survival mechanisms, would be overwhelmed within hours. If they land on exposed rock or in a desert, ultraviolet radiation becomes the immediate threat. Earth's ozone layer blocks the harshest UV wavelengths, but enough gets through to sterilize exposed surfaces. Bacteria deposited on sunlit rock or sand would be dead within a day. But if the particles land somewhere protected, in soil covered by vegetation, in the shadows of rocks, or in the perpetually dark deep ocean, they might have a chance to revive. The problem is competition. Earth's soil contains between 1 billion and 10 billion bacterial cells per gram. That's not a typo. 10 to the 9th or the 10th power organisms competing for the same resources in every handful of dirt. They've specialized into countless ecological roles. There are specialists for decomposing every type of organic matter. There are predators that hunt other bacteria. There are organisms that produce antibiotics specifically to kill competitors. It's biological warfare at the microscopic scale, and it's relentless. An alien bacterium would need extraordinary advantages to survive in that environment. It would need to metabolize something that Earth bacteria don't, or metabolize common compounds much more efficiently, or tolerate conditions that exclude native organisms. The first two options are unlikely because Earth bacteria have had 4 billion years to optimize for Earth chemistry. The third option, finding an unoccupied niche, is more plausible but requires the alien to tolerate conditions harsh enough that Earth bacteria haven't already colonized them. High radiation environments might offer one such niche. While Earth has radiation-resistant bacteria, they are relatively rare. Most of Earth's biosphere is shielded from intense radiation by atmosphere, water, or rock. A bacterium from 3I Atlas that evolved during billions of years of cosmic ray exposure might have radiation resistance capabilities exceeding anything Earth has produced. If such organisms landed near nuclear facilities, in uranium-rich geological formations, or in other high radiation environments, they might find a foothold. Extreme environments are another possibility. Earth has acidophiles that thrive at pH 2, more acidic than stomach acid. It has alkaliphiles that grow at pH 12, as caustic as bleach. But the specific metabolic strategies these organisms use evolved here, optimized for Earth's particular chemical conditions. A bacterium from 3I Atlas might have different strategies for handling pH extremes, strategies that work better in specific environmental contexts. Those contexts could provide niches where the alien could survive alongside natives rather than being outcompeted. The real danger from a planetary protection perspective isn't that alien bacteria would outcompete Earth life directly. It's that they would exchange genes with Earth bacteria. This is where horizontal gene transfer becomes a double-edged sword. If alien organisms carry genes that, too, provide novel capabilities, Earth bacteria that acquire those genes gain advantages. Most of the time, this is harmless or even beneficial. New metabolic pathways, improved stress tolerance. These are genetic innovations that could help Earth's biosphere adapt to changing conditions. But there are darker possibilities. Imagine an alien bacterium carrying genes for toxin production, compounds that affect eukaryotic cells in ways Earth organisms have never encountered. Earth bacteria that pick up those genes could become pathogenic to plants, animals, or humans, even if the original alien organism was harmless. Or consider genes that allow extreme environmental tolerance, enabling bacteria to survive in places they couldn't reach before. Industrial facilities, sterile medical environments, even clean rooms designed to exclude all contamination. Bacteria with such capabilities could become serious problems. The probability of any of this happening is low but non-zero. Most particles from 3I Atlas that reach Earth will be sterilized during entry or upon landing. Most that survive will be unable to compete with native organisms. Most that manage to exchange genes will transfer capabilities that are redundant with what Earth bacteria already possess. But most isn't all, and when we're dealing with billions of trillions of particles released by the comet, even extremely low probability events become possible. There's a precedent for concern here. Earth bacteria have been introduced to new continents multiple times in human history, often with devastating consequences. Diseases that evolved in one population suddenly encounter another with no immunity. Invasive species that have no natural predators in their new environment proliferate wildly. The biological equivalent of nuclear explosions can result from introducing just the wrong organism into an unprepared ecosystem. Now imagine that scenario. But instead of moving organisms between continents on the same planet, we're talking about moving them between worlds. The counter-argument is that Earth has been receiving extraterrestrial material for its entire history. Meteorites fall constantly. Comets deposit material regularly. If alien bacteria were going to cause problems, wouldn't they have done so already? The answer is yes, probably they would have, which suggests either that viable alien bacteria have never reached Earth in sufficient numbers to establish themselves, or that they have, but we haven't noticed, but the effects were subtle. There's a third possibility that's both reassuring and unsettling. Maybe alien bacteria have successfully colonized Earth multiple times throughout its history. Maybe some fraction of Earth's bacterial diversity doesn't descend from a single origin of life here. Maybe some lineages carry genetic information from other star systems, information that's been so thoroughly mixed with terrestrial genes through billions of years of horizontal transfer that we can't distinguish the original source. If true, then life on Earth is already a hybrid, already carrying the genetic legacy of multiple worlds. Adding a bit more from 3i Atlas wouldn't fundamentally change anything. But 3i Atlas is different from previous interstellar visitors because we're aware of it. We're watching it. We know it passed close to Mars and that its debris cloud intersects Earth's orbit. If there are consequences, if alien bacteria do establish themselves here, we might actually be able to detect it this time. Not immediately, perhaps not for years or decades, but eventually. We'd see novel genetic sequences appearing in bacterial populations. We'd see new metabolic capabilities spreading through microbial communities. We'd see evidence in the genomic record of a recent horizontal gene transfer event from sources that don't match any known terrestrial lineage. The search for these signatures requires sequencing bacterial genomes from diverse environments and looking for anomalies, genes that cluster together in ways that don't match standard phylogenetic relationships, metabolic pathways that use slightly different enzymes than the terrestrial equivalents, proteins with amino acid sequences that suggest optimization for different conditions than what exists on Earth. These would be subtle clues, easy to miss, but they'd be there if we knew how to look. There's also the possibility of more obvious effects. If alien bacteria carrying novel genes establish themselves in specific environments, those environments might change in detectable ways. Unusual algal blooms in water bodies, shifts in soil chemistry, changes in the dominant bacterial species in particular ecological niches. Environmental monitoring programs might pick up these signals without realizing their significance. A slight increase in a rare bacterial species here, an unexplained change in nutrient cycling rates there, individually meaningless, but collectively suggesting that something new has entered the system. The timeline for any of this to become apparent would be years to decades. Bacterial evolution operates on generational timescales. Even with rapid reproduction, it takes time for new genes to spread through populations, for selection to act, for measurable changes to accumulate. We wouldn't see sudden dramatic transformations. We'd see gradual shifts, trends that only become clear when you compare data from before and after the 3i Atlas passage. This is where citizen science could play a role. Amateur microbiologists sampling and sequencing bacteria from their local environments, distributed monitoring projects tracking changes in microbial communities across wide geographic areas, databases where people can upload sequence data and have it analysed for anomalies. The more eyes watching, the more likely we are to catch something unusual if it appears. The reality is that we probably won't know whether bacteria from 3i Atlas successfully colonised Earth unless they cause obvious problems, or unless we get lucky with genetic sequencing. The microbial world is vast and mostly unexplored. We've sequenced perhaps 1% of bacterial species on Earth. The rest remain unknown, their genes unread, their capabilities uncharacterised. Against that background of ignorance, detecting the introduction of alien organisms is like finding specific needles in a planet-sized haystack. Possible, but requiring dedicated effort, sophisticated tools and considerable luck. What we can say is that Earth's uninvited guests, if they're here, are facing the same challenges that any invasive species faces. They need to survive initial conditions that are hostile to them. They need to find resources. They need to reproduce faster than they die. And they need to do all of this while competing with organisms that are already perfectly adapted to local conditions. It's not impossible, but it's difficult. Which is probably for the best, because a universe where biological contamination between worlds was easy would be a universe where planet-scale ecological disasters were inevitable and common. The acceleration problem: evolution is supposed to be slow. Natural selection operating on random mutations generation after generation, gradually accumulating beneficial changes while purging harmful ones. Darwin's vision of descent with modification proceeding at rates measured in millions of years. Except bacteria don't follow those rules. They demonstrate that evolution can operate in fast forward when the conditions are right, and the 3i Atlas scenario creates exactly those conditions on both Mars and Earth. The traditional model of bacterial evolution assumes isolated populations changing independently through mutation and the selection. The rates are predictable. A typical bacterial species might accumulate one beneficial mutation per million generations under standard laboratory conditions. In nature, where selective pressures are more intense and populations larger, the rate might increase to 1 per 100,000 generations. Still slow by human standards, still requiring vast timescales to produce major changes, but that model collapses when horizontal gene transfer enters the picture. Studies tracking gene transfer in bacterial populations have revealed something startling. In environments where multiple bacterial lineages encounter each other, beneficial genes can spread through the entire population in under 1,000 generations, not by waiting for the right mutation to arise independently, but by copying genes that already exist in another organism. The difference between these timescales is the difference between waiting for evolution and hijacking it. On Mars, if bacteria from 3I Atlas encounter any native Martian life, or even if they encounter other lineages of bacteria from previous cometary deliveries, we have the setup for accelerated evolution: multiple genetic toolkits converging in the same environment, multiple solutions to similar problems available for mixing and matching. The organisms don't have to reinvent radiation resistance or cold tolerance or desiccation resistance. They can acquire those capabilities wholesale from neighbours who already have them. The acceleration isn't just about speed, it's about the solution space that becomes accessible. Imagine you're trying to navigate a maze. The mutation-only approach is like walking randomly, hoping to stumble on the exit. Horizontal gene transfer is like having access to a map showing paths that worked for other travellers. You can skip dead ends entirely. You can jump to successful routes without having to discover them through trial and error. Laboratory experiments have quantified this effect. Researchers evolve bacterial populations in challenging environments with and without access to foreign DNA. Populations with DNA access consistently find adaptive solutions 5 to 10 times faster than populations relying solely on mutation. In some cases, populations without access to foreign DNA fail to adapt at all within the experimental timeframe, while populations with access thrive. The presence or absence of horizontal gene transfer doesn't just change the rate of evolution, it changes whether evolution succeeds. For bacteria from 3I Atlas landing on Mars or Earth, this has profound implications. Even if the organisms are poorly adapted initially, even if 99.99% of them die, the tiny fraction that survives has access to 4 billion years of terrestrial genetic innovation if they reach Earth, or potentially billions of years of Martian innovation if native life exists there. Those survivors can sample that genetic library through horizontal transfer, testing different combinations until they find something that works. The process creates a form of evolutionary ratchet. Each successful gene transfer produces organisms that are slightly better adapted. Those better adapted organisms survive longer, produce more offspring, and have more opportunities to acquire additional beneficial genes. The effect compounds. Within a few hundred generations, you can see dramatic shifts in population genetics. Within a few thousand generations, organisms might be so changed that they'd be unrecognizable compared to their ancestors. But there's a problem, and it's one that evolutionary biologists are only beginning to understand. Rapid horizontal gene transfer can create mismatches. Genes that work well in one genetic background might function poorly in another. Regulatory networks get disrupted, protein interactions fail, the organism acquires a theoretically beneficial gene, but can't actually use it because the rest of its cellular machinery isn't compatible. This is where the acceleration problem becomes apparent. Evolution through mutation and selection is slow, but it's coherent. Changes accumulate in ways that maintain functional integration. The organism stays viable at every step because selection eliminates any changes that break essential systems. Horizontal gene transfer can be so fast that it outpaces the mechanisms for maintaining coherence. Organisms acquire genes faster than they can optimize their genomes to accommodate them. The result is something called genetic load, the burden of carrying genes that are neutral or slightly harmful but haven't been purged yet. In stable environments, natural selection gradually cleans this up. Organisms with lower genetic load outcompete organisms with higher load. But in rapidly changing environments, especially harsh environments where most organisms are stressed, selection becomes less efficient. Slightly harmful genes can persist because organisms carrying them happen to also carry other genes that provide larger benefits. The population tolerates some dysfunction because the alternative is extinction. On Mars, bacterial populations would almost certainly accumulate high genetic loads. The environment is so marginal that any organism capable of surviving at all would be retained by selection regardless of how many suboptimal genes it carries. Horizontal transfer would introduce genes at rates far exceeding what natural selection could efficiently evaluate. The result would be genomes full of redundant, partly broken, or poorly integrated genetic elements. These organisms would work, but they wouldn't be elegant. They'd be evolutionary jury rigs cobbled together from whatever pieces were available. This has implications for how fast Martian bacterial evolution could proceed. On one hand, horizontal transfer accelerates the initial adaptation to Martian conditions. On the other hand, the accumulation of genetic load slows subsequent adaptation because the organisms are carrying so much genomic baggage. There's a balance point, a rate of gene transfer that's fast enough to enable survival, but not so fast that it overwhelms the organism's ability to integrate new genes functionally. Earth bacteria encountering genes from 3I Atlas face a different version of the same problem. Earth's bacterial populations are highly optimized for their environments. They've had billions of years to refine their genomes. Introduction of foreign genes, even beneficial ones, disrupts that optimization. The immediate benefit from a new metabolic capability might be offset by the costs of integrating that capability into existing regulatory networks. This is why most horizontal gene transfer events are neutral or slightly deleterious. Only a small fraction provides enough benefit to overcome the integration costs. But that small fraction is what matters for evolution. Those rare successful transfers where a gene provides a large enough advantage that the integration costs are worth paying are the ones that spread through populations and produce lasting change. There's a temporal aspect to this that's crucial. In the immediate aftermath of 3I Atlas passing Mars, any bacteria successfully delivered would be under intense selective pressure. Survival trumps everything else. Organisms would acquire genes promiscuously, taking anything that might help. Most of those acquisitions would be useless or harmful, but some would be life-saving. The population would experience a burst of rapid evolution driven by the desperate scramble to adapt. Then, assuming some organisms do adapt and start reproducing, the pace would slow. Selection would begin cleaning up the genomic mess accumulated during the crisis. Redundant genes would be lost. Regulatory networks would be streamlined. Integration problems would be resolved through compensatory mutations. The population would transition from rapid, messy adaptation to slower, more refined optimization. This is the pattern seen in laboratory evolution experiments when bacteria are suddenly exposed to harsh new environments. The timescale for this transition depends on generation time. On Mars, where bacterial reproduction might take weeks or months per generation due to extreme cold and nutrient limitation, the whole process would be slower than on Earth. But it would still be fast by geological standards. Within a few thousand to million years, you'd have bacterial populations fully adapted to Martian conditions, their genomes stabilized, their metabolic networks integrated and efficient. On Earth, the timescale would be much faster. Bacteria with generation times measured in hours or days could go through the entire cycle of rapid adaptation, followed by optimization in decades to centuries. If bacteria from 3I Atlas did successfully colonize Earth niches, we might be able to watch the process happen in real time, or at least on timescales short enough for human observation. There's another layer to the acceleration problem that's both fascinating and concerning. Horizontal gene transfer doesn't just accelerate evolution within bacterial populations, it can accelerate it across domains of life. Bacteria can transfer genes to archaea, to fungi, even to plants and animals. The transfers are rare, but they happen, and when they do, they can have dramatic effects. If bacteria from 3I Atlas carry genes that are functionally compatible with eukaryotic systems, those genes could theoretically transfer to Earth's plants, animals, or fungi. The probability is extremely low. The barriers to cross-domain gene transfer are significant. But low probability isn't zero probability, and we're dealing with astronomical numbers of potential transfer events. A 1 in a billion chance becomes meaningful when you're rolling the dice trillions of times. The most likely scenario for cross-domain transfer would involve bacteria that form symbiotic relationships with eukaryotes. Many organisms host bacterial symbionts that provide essential metabolic functions. These relationships create opportunities for us. Gene transfer because the bacteria live inside host cells where their DNA is exposed to the host's genetic machinery. Over evolutionary time, genes have moved from bacterial symbionts into host genomes, becoming permanent parts of the eukaryotic genetic repertoire. Mitochondria and chloroplasts are the most famous examples, but there are many others. If bacteria carrying genes from 3I Atlas establish themselves as symbionts in Earth organisms, those genes could potentially integrate into host genomes. The functions they provide would need to be beneficial to the host for selection to retain them, but that's not impossible. A gene for enhanced radiation resistance might help organisms colonize high-altitude or high-radiation environments. A gene for metabolizing novel compounds might allow access to new food sources. The scenarios are speculative, but they're not physically impossible. What makes the acceleration problem genuinely problematic is that we can't predict the outcomes. Evolution in fast-forward mode explores genetic space rapidly and often chaotically. It finds solutions that slow evolution would never discover, because those solutions require multiple simultaneous changes that are individually neutral or harmful. When horizontal gene transfer makes those simultaneous changes possible, you get evolutionary trajectories that look discontinuous, almost like jumps, rather than gradual transitions. This is exciting scientifically because it suggests life can adapt to new conditions much faster than classical evolutionary theory predicts. It's concerning from a planetary protection standpoint because it means biological contamination events might have consequences that manifest faster and in more unpredictable ways than we've assumed. The safe timeline we thought we had, millions of years for any introduced organisms to become problematic, might collapse to thousands of years or less if horizontal transfer is rampant. The acceleration problem doesn't have a solution because it's not really a problem in the engineering sense. It's a feature of how evolution works when genetic information can flow freely between organisms. We can't slow it down without fundamentally altering the nature of bacterial life. What we can do is monitor for it, watch for the signatures of rapid horizontal transfer in natural populations, and try to understand the changes as they happen. Whether those changes will ultimately be beneficial, harmful, or neutral is something we'll only know after they've occurred. When worlds collide with biology, the concept of biological collision sounds violent, but at the microscopic level it's more subtle. Two separate evolutionary lineages, products of completely different planetary histories meeting in the same environment. The question isn't whether they fight or cooperate. The question is whether they can coexist at all, and if so, what emerges from that coexistence. Consider what it means for a bacterium from 3I Atlas to encounter a Martian organism, assuming Martian life exists. These two lineages would have had no contact since they diverged, possibly billions of years ago if both trace back to a common origin, or Nevirus if they arose independently. Their biochemistry would have been shaped by different planetary conditions, different atmospheric compositions, different available minerals, different radiation environments, different selective pressures at every level from molecular to ecological. Yet if both lineages use DNA as genetic material, if both use proteins as functional molecules, if both require liquid water as a solvent, they share fundamental compatibility. Their genetic codes might differ in detail but work on the same principles. Their metabolic pathways might use different enzymes but accomplish similar chemical transformations. The overlap creates opportunity for interaction and for horizontal gene transfer, but the differences create potential for conflict and incompatibility. Biochemical mismatch is the first challenge. Earth organisms use specific amino acids, specific nucleotides, specific lipids in their cellular structures. These aren't arbitrary choices. They're optimized solutions that emerged from 4 billion years of evolution in Earth's particular chemical environment. Change the environment, change the available building blocks, and evolution might optimize for different solutions. Bacteria from 3I Atlas might use a slightly different set of amino acids. They might prefer different membrane lipids. These differences could prevent successful gene transfer even if the organisms encounter each other. But biochemistry is flexible. Bacteria are known to be able to incorporate non-standard amino acids into their proteins when faced with nutrient limitations. They can modify their membranes to function in radically different conditions. The ability to adapt, to work around mismatches, is built into the fundamental nature of bacterial life. So while biochemical differences create barriers, they're not absolute walls, They're more like filters that allow some interactions while blocking others. Metabolic compatibility is another layer. Two bacteria might be able to exchange genes successfully, but be unable to coexist in the same microenvironment because they compete for identical resources. This is Gauss's competitive exclusion principle in action. Two species cannot indefinitely occupy the same ecological niche if resources are limited. One will always outcompete the other. For bacteria from 3I Atlas to coexist with native Martian organisms, they need to partition resources somehow, specialize in different substrates, tolerate different stress levels, or occupy different spatial niches within the subsurface. This is where the 3I Atlas scenario becomes particularly interesting. The bacteria arriving from the comet wouldn't be a single species. Different regions of the comet's interior would host different organisms adapted to different microenvironments within the ice. Some might specialize in extremely cold conditions. Others might be adapted to brief periods of warmth near the comet's surface when it approaches other stars. Still others might tolerate high radiation doses or extreme desiccation. This diversity means the comet delivers a whole suite of potential colonizers, each with different capabilities. When that diverse community arrives on Mars and encounters potentially diverse native life, you don't get one collision. You get dozens or hundreds and trids of separate collisions, each with different outcomes. Some alien species might be completely incompatible with Martian conditions and die immediately. Others might find unoccupied niches and thrive without ever encountering natives. Still others might compete directly with Martian organisms, leading to displacement or coexistence depending on the details of their metabolic strategies. The pattern we'd expect to see is something ecologists call community assembly. Different species sorting themselves across the available environmental gradients, each finding the combination of conditions where it performs best. In some locations, alien bacteria might dominate. In others, natives would exclude the newcomers. In still others, you would have mixed communities where both lineages coexist because they use resources in complementary rather than competing ways. Over time, horizontal gene transfer would blur these distinctions. Alien bacteria acquiring genes from natives become more native-like. Natives acquiring genes from aliens become more cosmopolitan. The genetic boundaries between lineages erode, replaced by a continuum of hybrid organisms carrying genes from both origins. This process, called genetic homogenization, is seen in Earth bacteria when previously isolated populations come into contact. Given enough time, they merge into what's functionally a single gene pool, even if they maintain separate identities at the species level. On Earth, the collision dynamics would be different, because Earth's biosphere is so much more competitive. Bacteria from 3I Atlas wouldn't encounter empty niches waiting to be filled. They'd encounter environments already packed with organisms using every available resource. The only way aliens could establish themselves would be to bring something novel, some capability that Earth bacteria lack entirely, or perform less efficiently. One possibility is radiation resistance. While Earth has radiation-resistant bacteria, they evolved that capability relatively recently, in evolutionary terms, and primarily in response to human activities like nuclear technology. Natural high radiation environments on Earth are rare. A bacterium from 3I Atlas that spent billions of years bathed in cosmic rays might have radiation resistance mechanisms far exceeding anything Earth has produced. Those mechanisms, encoded in specific genes, could transfer to Earth bacteria and spread through populations exposed to radiation. Another possibility is metabolic versatility. Many extremophile bacteria on Earth are specialists, highly adapted to specific extreme conditions but unable to function outside those conditions. A bacterium from 3I Atlas might be a generalist, capable of tolerating a wider range of conditions because it had to cope with the varying environments it encountered during its interstellar journey. That versatility, even if it comes at the cost of not being optimal in any single environment, could be valuable in Earth's highly variable habitats. The collision also operates at the viral level. If 3I Atlas carries bacteriophages, viruses that infect bacteria, those phages arrive along with their hosts. Phages are major players in bacterial evolution, driving horizontal gene transfer, controlling population densities, and serving as genetic reservoirs. A phage from another star system encountering Earth bacteria is essentially a genetic warfare scenario. Can the phage infect Earth bacteria? If so, it could kill them, transfer genes to them, or incorporate itself into their genomes as a prophage. Most likely, phages from 3I Atlas wouldn't be able to infect Earth bacteria because the molecular recognition systems that allow phages to attach to host cells are highly specific. But if even a small fraction of alien phages can cross that barrier, they can have dramatic effects. Phage-mediated gene transfer is incredibly efficient. A single successful infection can convert a bacterium into a factory producing thousands of viral copies, each potentially carrying genes from previous hosts. This amplifies the spread of genetic information far beyond what direct cell-to-cell transfer could achieve. The temporal dynamics of biological collision matter enormously. In the short term, immediate competition and rapid horizontal transfer dominate. Organisms scramble to adapt or die. Gene exchanges are frequent and chaotic. Population sizes fluctuate wildly. This is the crisis phase, where outcomes are highly uncertain and small differences in initial conditions can lead to radically different results. In the medium term, things stabilize. Surviving lineages establish themselves in specific niches. Gene transfer continues but becomes more selective, focusing on genes that provide clear benefits rather than random sampling. Communities develop structure, with different species occupying different roles. This is the organization phase, where the chaos resolves into patterns. In the long term, the distinction between native and alien disappears. Genomes have exchanged so many genes that tracking origins becomes impossible. The community functions as an integrated whole, with organisms carrying genetic information from multiple sources, blended through billions of years of mixing. This is the integration phase, where separate evolutionary histories merge into a unified biological system. Earth has probably been through this process multiple times already if panspermia is real. Every major cometary impact could potentially deliver microorganisms from other environments. Most introductions probably fail, but over 4 billion years, some must have succeeded. The genetic legacy of those ancient collisions might still be present in Earth's bacterial genomes, hiding in plain sight because we don't know how to recognize it. Genes we attribute to Earth's evolutionary history might actually have origins elsewhere, acquired so long ago that the signal of their introduction has been erased by subsequent evolution. 3I Atlas represents a collision we can potentially track. We know when it happened. We can watch for genetic signatures appearing in bacterial populations after the encounter. We might even be able to identify specific genes that don't fit standard phylogenetic patterns, genes that cluster with each other, but not with any known terrestrial lineages. These would be the smoking guns, direct evidence of recent horizontal transfer from an extraterrestrial source. The challenge is distinguishing those signals from noise. Bacterial genomes are full of weird genes acquired through ancient horizontal transfers from unknown sources. Showing that a particular gene came from 3I Atlas rather than some other historical source requires strong evidence. Timing is crucial. If we see genes appearing in populations that weren't present before the comet's passage, that's suggestive. If we see those genes spreading rapidly through communities, that's more suggestive. If we can match their sequences to known extremophile capabilities consistent with survival in interstellar space, that's even stronger. But none of this constitutes proof. Proof would require isolating organisms, culturing them, sequencing their genomes completely, and demonstrating beyond doubt that they carry genetic information inconsistent with terrestrial origin. That's a high bar. It might be too high to ever clear without more sophisticated techniques than we currently possess. What we can say with confidence is that biological collisions, when they occur, have the potential to reshape entire ecosystems. The introduction of a single successful lineage can cascade through food webs, altering nutrient cycles, changing competitive relationships, and opening new evolutionary possibilities. If bacteria from 3I Atlas successfully colonized Mars or Earth, we're not talking about a minor footnote in biological history. We're talking about a potentially planet-scale event with consequences that might take millions of years to fully unfold. The collision between worlds isn't just about bacteria meeting each other. It's about different solutions to the problem of life encountering each other, testing which solutions work better under which conditions, and creating hybrid systems that combine the best of both. It's evolution operating on a cosmic scale using entire planetary biospheres as laboratories and comets as vectors for exchanging results. When worlds collide biologically, the outcome isn't destruction. It's creation, the emergence of something new that couldn't have existed in isolation. The Future Martian Biosphere Project Forward. Not years, but millennia. Not thousands of years, but millions. If bacteria from 3I Atlas successfully colonized Mars's subsurface, what would that biosphere look like as it matures and evolves? This isn't science fiction speculation. It's applying what we know about bacterial evolution, ecology, and planetary science to predict plausible outcomes. The starting point is a sparse population of bacteria distributed unevenly through Martian subsurface ice, rock fractures, and possibly aquifers beneath the polar caps. These organisms are stressed, operating at the absolute limits of what biology can tolerate. Growth rates are extraordinarily slow. Reproduction might occur once per year, once per decade, or even once per century, depending on how cold and nutrient-poor the specific environment is. At these rates, even 1,000,000 years represents only 10,000 to 1,000,000 generations. For bacteria, that's enough time for significant evolution, but not enough for complete transformation. The first major evolutionary pressure would be optimization for the cold. Earth's coldest adapted bacteria, psychrophiles, can grow at temperatures down to -15 degrees Celsius. Below that, metabolism essentially stops in pure water. But Martian subsurface environments don't contain pure water. They contain brines, salt-saturated solutions that remain liquid at much lower temperatures. Bacteria that can function in these brines would have a massive advantage. Evolution toward cold adaptation involves multiple changes. Enzymes need to remain flexible at low temperatures, while most proteins become rigid. Membranes need to stay fluid when ice crystals would normally freeze them solid. DNA repair mechanisms need to function despite the fact that chemical reactions proceed millions of times slower than at room temperature. Each of these adaptations requires genetic changes, multiple mutations coordinating to shift the organism's entire physiology toward cold tolerance. If bacteria from 3I Atlas brought genes for extreme cold tolerance from their home system, those genes would spread rapidly through any Martian bacterial population through horizontal transfer. Within a few thousand generations, you'd expect to see those genes in most or all lineages that survived. The population would become collectively more cold tolerant, not through each lineage independently evolving the same solutions, but through sharing the best solutions discovered by any member of the community. The second pressure would be radiation management. Even underground, Martian bacteria face constant bombardment from cosmic rays and radioactive decay of surrounding rocks. Over millions of years, the accumulated radiation damage would be enormous. Only organisms with robust DNA repair mechanisms could persist. Again, if bacteria from 3I Atlas carried superior radiation resistance genes from their journey between stars, those genes would be valuable currency in the Martian gene pool. But there's a twist. Radiation creates reactive oxygen species, harmful molecules that damage cellular components. On Earth, aerobic bacteria evolved elaborate systems for dealing with oxidative stress because they live in an oxygen-rich atmosphere. Mars's atmosphere is mostly carbon dioxide with almost no free oxygen. Martian bacteria wouldn't face oxidative stress from the atmosphere, only from radiation-induced reactions. They might evolve completely different approaches to managing oxidative damage. Approaches optimized for low oxygen, high radiation conditions rather than the high oxygen, low radiation conditions that shaped Earth bacteria. The third pressure would be nutrient scarcity. Mars is a nutrient-poor environment. Nitrogen is rare. Phosphorus is locked in minerals that dissolve slowly. Trace elements essential for life, like iron, zinc, and molybdenum, are present, but not necessarily accessible. Bacteria capable of efficiently scavenging nutrients from their environment would outcompete less efficient scavengers. This drives evolution toward improved uptake systems, better mineral-dissolving enzymes, and more effective nutrient recycling. Over millions of years, these selection pressures would sculpt the Martian bacterial community into something distinctly non-terrestrial. The organisms would still be recognizable as bacteria, still use DNA and proteins and cellular membranes, but their specific adaptations would be unique to Mars. They'd be optimized for cold brines, high radiation, and extreme nutrient limitation in ways Earth bacteria never needed to be. The ecological structure would be simple compared to Earth. No photosynthesis because there's no light underground. No oxygen-based respiration because there's no atmospheric oxygen. The entire ecosystem would run on chemosynthesis, bacteria extracting energy from chemical reactions between minerals and dissolved gases. Hydrogen oxidation would be the primary energy source. Carbon dioxide reduction would be the primary carbon-fixing pathway. Sulfur and iron cycling would connect different metabolic guilds. But simple doesn't mean static. Within those constraints, diversification could occur. Different lineages might specialize in different chemical reactions. Some might focus on hydrogen oxidation coupled to sulfate reduction. Others might use iron reduction, or even perchlorate reduction. Still others might act as fermenters, breaking down organic molecules produced by the primary producers. These different metabolic strategies would create ecological structure, different species occupying different biogeochemical niches. Spatial structure would emerge as well. Bacteria near geothermal vents, where subsurface heat keeps water liquid at higher temperatures, would form different communities than bacteria in the cold polar ice. Organisms in porous sandstone would face different challenges than organisms in dense basalt. Communities in sulfate-rich evaporite deposits would differ from communities in iron-rich volcanic rocks. Each environment would select for slightly different traits, creating geographic variation across Mars' subsurface. The most intriguing possibility is the emergence of multicellularity. Earth bacteria occasionally form multicellular structures like biofilms and fruiting bodies. These structures allow collective behavior, resource sharing, and division of labor. In extremely nutrient-poor environments like deep Earth subsurface, bacteria sometimes form long filaments connecting individual cells, possibly to facilitate nutrient distribution across large distances. Martian bacteria facing similar extreme nutrient limitation might evolve similar or even more elaborate multicellular structures. Imagine networks of bacterial filaments spreading through rock fractures, connecting cells in warm, nutrient-rich zones with cells in cold, nutrient-poor zones. The network as a whole could be more resilient than individual cells, buffering against local environmental fluctuations and distributing resources where they're needed most. This wouldn't be multicellularity in the animal or plant sense, but it would represent a level of organization beyond individual cells responding independently to their environment. The temporal scale for these developments is as uncertain. How fast could a Martian bacterial ecosystem evolve from the initial colonization by 3I Atlas to a mature, diversified biosphere? On Earth, bacterial ecosystems can reach equilibrium relatively quickly, but that's in environments with abundant resources and relatively favorable conditions. On Mars, everything happens slower. Cell division is slower. Selection acts more weakly. Opportunities for horizontal gene transfer are less frequent because population densities are lower. A reasonable estimate might be millions to tens of millions of years for significant diversification. For the emergence of truly novel adaptations not present in the founding population, perhaps 100 million years. These are long timescales by human standards, but brief in geological terms. Mars is 4.5 billion years old. If life has been attempting to establish itself there repeatedly through cometary delivery over that entire period, there's been plenty of time for sophisticated bacterial ecosystems to evolve, even under the harsh conditions. This raises a disturbing possibility. What if sophisticated Martian bacterial ecosystems already exist? What if the 3I Atlas delivery isn't the first successful colonization, but just the latest in a series stretching back billions of years? Native Martian life might already be diverse, already adapted, already filling the available niches in the subsurface. In that case, bacteria from 3I Atlas would be newcomers entering an established ecosystem, not pioneers colonizing an empty world. The implications for detection are significant. If Martian life is sparse, scattered, and confined to deep subsurface environments, our current exploration methods will miss it. The rovers can only scratch the surface, drilling at most a few meters down. The orbiters can only see what's on top or detect signatures from the upper layers. To find deep biospheres, we need dedicated missions with deep drilling capabilities, subsurface radar designed to detect water and organic signatures, and sample return missions targeting the most likely habitats. Several locations on Mars stand out as priorities. The subsurface beneath the southern polar ice cap, where liquid water has been detected. the deep aquifer systems possibly connected to ancient river valleys, the regions showing recent fluid flow in gullies and recurrent slope lineae, geothermally active areas where volcanic heat might maintain liquid water conditions. Each of these environments could harbour bacterial communities, either native or introduced, adapted to the specific local conditions. The search strategy should focus on biosignatures that would persist even if the organisms themselves are rare or dormant. Lipids from bacterial membranes can be preserved in rocks for billions of years. Isotopic signatures from metabolic fractionation remain detectable long after the organisms die. Minerals like magnetite or sulfides that bacteria produce through metabolism can indicate past biological activity. DNA fragments, if protected in ice or mineral matrices, might persist long enough for detection even without viable cells. But the ultimate goal isn't just finding evidence of life, it's understanding the biosphere as a system. What organisms are present? How are they related to each other and to Earth life? What metabolic strategies do they use? How do they interact with their environment? A truly mature Martian biosphere would be a complex web of metabolic interdependencies, different species relying on each other's waste products, creating feedback loops that stabilize the system against environmental perturbations. On Earth, we've learned that microbial ecosystems are far more complex than they appear. What looks like a simple community of a few species turns out, upon detailed genetic analysis, to be hundreds or thousands of distinct lineages interacting in intricate ways. Martian bacterial communities might display similar hidden complexity. Surface observations might show nothing but sterile rock, while meters below, a rich ecosystem thrives unseen. The future Martian biosphere, if it develops from 3I Atlas colonization or already exists from previous introductions, represents something unique in the known universe. It would be life adapted to conditions at the absolute edge of habitability, surviving through strategies we can barely imagine, creating an ecosystem functioning on timescales that make human lifespans irrelevant. It would be a laboratory for understanding how far biology can be pushed, what universal principles govern life regardless of environment, and what diversity is possible within the constraints of physics and chemistry. And someday, if humans establish a permanent presence on Mars, we'll have to decide how to interact with this biosphere. Do we protect it, keep it pristine for scientific study? Do we ignore it, focusing on surface habitats where it doesn't exist? Do we try to enhance it, deliberately introducing organisms that might help terraform the planet? These are not just scientific questions. They are ethical questions about our responsibilities to other forms of life and about what we want Mars to become. The future Martian biosphere might be invisible now, but its foundations could be settling into place as debris from 3I Atlas drifts down through the thin atmosphere and begins the slow process of adaptation. In 1 million years, Mars might host a rich subsurface ecosystem. In 10 million years, it might be teeming with bacterial life cycling elements, processing minerals and quietly conducting the business of survival in one of the solar system's harshest environments. And all of it would trace back to a comet that happened to pass by carrying passengers from another star. What this means for us, we've travelled through a hypothesis, following bacteria from an interstellar comet as they potentially seed 2 worlds with life from another star system. Now we need to step back and ask the uncomfortable question. If any of this is true, what does it mean for us? Not just as scientists or theorists, but as a species that will soon be exploring Mars directly. The first implication is philosophical. If life on Earth contains genetic information from other star systems, delivered by comets over billions of years, then our origin story is more complicated than we thought. We're not purely products of Earth's environment. We're hybrids, carrying biological heritage from multiple worlds blended together over cosmic timescales. Every organism on Earth, including us, might contain genes that originated elsewhere, so thoroughly integrated into our biology that we can't distinguish them from terrestrial innovations. This challenges the narrative of human uniqueness. We like to think of ourselves as special, the pinnacle of Earth's 4 billion year evolutionary experiment. But if Earth's biosphere has been regularly supplemented by genetic material from other systems, then we're not pure products of local evolution. We're part of a galactic ecosystem. One world among many contributing to and drawing from a shared pool of biological information distributed by comets and asteroids throughout the galaxy. Some people find this idea unsettling. It undermines the sense that Earth is our special home, that we emerged from this specific place through a unique sequence of events. But I think it's actually more profound. It means we're connected to the larger universe in a fundamental biological way, not just through physics and chemistry, but through the actual genetic code that built us. We're not alone in splendid isolation. We're participants in a cosmic exchange of life that's been ongoing since stars first formed. The second implication is practical and urgent. If bacteria from 3I Atlas did reach Mars and are currently adapting to subsurface conditions, then Mars is not a sterile world. It's a living world, even if that life is invisible and confined to deep environments. This changes everything about how we should approach Mars exploration and eventual human settlement. Current planetary protection protocols are based on the assumption that Mars might have native life, but that assumption is theoretical. We have no proof Martian life exists. The protocols are precautionary, designed to prevent contamination of a potentially living world. But if 3I Atlas delivered viable bacteria just months ago, then we're not protecting a potential biosphere anymore. We're protecting an actual biosphere, one that's in the early stages of establishment. This creates ethical dilemmas. Do we have the right to interfere with this process? If bacteria from elsewhere are trying to colonize Mars, should we help them by deliberately introducing terrestrial organisms that might share useful genes through horizontal transfer? Should we hinder them by sterilizing areas where we plan to conduct human activities? Or should we simply observe, keeping our distance and letting nature take its course? There's no consensus on these questions because we've never faced them before. Every discussion of planetary protection has been hypothetical, what we would do if we found life. Now we might be in a situation where life was recently introduced, is potentially establishing itself, and we're about to mount much more intensive exploration of exactly the environments where that life would be found. The decisions we make in the next few decades could determine whether Mars develops a robust biosphere or remains essentially sterile. The third implication concerns human health and Earth's biosphere. If particles from 3I Atlas reached Earth and contained viable bacteria, those organisms are here now. Probably they died or are struggling in marginal environments unable to compete with native life. But possibly not. Possibly some found niches where they can survive. Possibly they're exchanging genes with Earth bacteria. Possibly we're already living alongside organisms carrying genetic information from another star system and don't know it. This isn't a disaster scenario. It's not an alien invasion. Most horizontal gene transfer events are neutral or beneficial, but we should be monitoring for unusual changes in bacterial populations, especially in extreme environments where novel organisms would be most likely to establish themselves. High radiation facilities, geothermal systems, deep ocean vents, Antarctic ice. These are the places where bacteria from 3I Atlas might find refuge, and these are the places we should watch most carefully. The monitoring doesn't require new infrastructure. We already have environmental genomic sequencing programs, citizen science initiatives, and databases tracking bacterial diversity. What we need is awareness that we should be looking for specific signatures. Genes that don't fit standard phylogenetic trees. Metabolic capabilities appearing suddenly in populations that didn't have them before. Clusters of related sequences showing up simultaneously in geographically distant locations. These would be red flags suggesting recent horizontal transfer from an external source. If we do find evidence of bacterial colonization from 3I Atlas, the response should be measured. Not panic, not overreaction, but systematic study. Isolate the organisms if possible. Sequence their genomes. Determine what capabilities they carry and whether those capabilities pose any risks. Most likely they'll turn out to be harmless, just another set of bacterial species joining Earth's already enormous diversity. But we should verify that rather than assume it. The fourth implication is about our future as a spacefaring species. If panspermia is real, if life spreads naturally between worlds via comets and asteroid beds, then we need to accept that perfect planetary protection is impossible. We can minimize contamination. We can be careful with our spacecraft and samples. But we can't prevent all transfer of organisms between worlds because nature is already doing it constantly on timescales of millions of years. This doesn't mean we should be reckless. It means our approach to planetary protection should be pragmatic rather than absolutist. protect Mars from massive contamination that could overwhelm any existing biosphere, prevent introduction of organisms specifically adapted to outcompete native life, but accept that small-scale exchanges are natural and have been happening throughout the solar system's history. Our spacecraft might carry a few thousand bacterial spores. A single comet can carry trillions. The scales aren't comparable. What we should focus on is understanding, not just preventing. If we're going to be a spacefaring civilization, we need to understand how biology behaves in space, how organisms adapt to different planetary environments, and how ecosystems develop on worlds with different conditions than Earth. This means studying natural experiments like the 3-Eye Atlas scenario, tracking what happens when organisms are delivered to new worlds, and learning from both successes and failures. Mars will likely become a test case for these questions. If we establish permanent human presence there, we will inevitably introduce Earth organisms. Bacteria will escape from habitats no matter how good our containment is. Plants in greenhouses will release seeds. Water recycling systems will leak microbes. Over decades and centuries, Mars will become contaminated with terrestrial life whether we intend it or not. The question isn't whether this will happen. The question is whether we manage it intelligently or stumble into it blindly. Intelligent management means preserving some regions of Mars as biological reserves, areas where we don't go and don't contaminate, allowing us to continue searching for native or naturally introduced life without overwhelming it with organisms we brought deliberately. It means careful monitoring of how introduced organisms behave in Martian conditions, what adaptations they develop, and whether they spread beyond our habitats. It means being prepared to respond if introduced organisms cause unexpected problems, though frankly that's unlikely given how harsh Mars is. The fifth and final implication is about perspective. The story we've explored, bacteria from 3-Eye Atlas potentially seeding Mars and Earth with life from another star, is just one example of a process that's probably ubiquitous throughout the galaxy. Every star system with comets is potentially exchanging life with every other system. Over billions of years, this creates a web of biological connection, genetic information flowing between worlds on cometary vectors, mixing and remixing into countless combinations. Life in the galaxy might not be isolated islands, each world developing independently. It might be more like an archipelago, with occasional connections between islands allowing exchange of species and genes. Some islands might be sources, pumping out organisms that colonize other worlds. Some might be sinks, receiving but not exporting. Some might be both, participating fully in the galactic ecosystem. Earth could be any of these. We might be primarily a source, our bacteria hitching rides on debris knocked off the planet by asteroid impacts, spreading to other worlds. We might be primarily a sink, receiving more than we send because our rich biosphere makes us an attractive target for colonization. Most likely we're both simultaneously sending and receiving part of the great cosmic exchange. This means that when we search for life elsewhere, we should expect to find biochemistry similar to ours, not just because similar chemistry leads to similar solutions, but because we might literally share genetic heritage with life on other worlds. The universal genetic code might be universal not because it's the only possible solution, but because it's spread from world to world, becoming standard through the same kind of network effects that make certain technologies dominant even when alternatives exist. It also means we're not alone, even if we never meet intelligent aliens. We're connected biologically to other worlds through the bacteria we carry, the genes we've inherited from cometary deliveries, and the organisms will inevitably spread when we explore and settle other planets. Every human contains within their microbiome bacteria descended from lineages that might trace back to other star systems. Every plant, every animal, every organism on Earth is part of this larger story. The 3-Eye Atlas scenario isn't just about whether a comet delivered bacteria to Mars and Earth. It's about recognizing that we're part of a living galaxy, that biological information flows between worlds just as matter and energy do, and that life's ability to adapt and spread makes it a cosmic phenomenon, not a planetary one. We emerged from this process. We participate in it. And as we expand into space, we become agents of it, deliberately and accidentally spreading Earth's biology to new worlds just as other worlds may have spread their biology to us. What this means for us is that we need to think bigger, not just about life on Earth or life on Mars, but about life as a galactic process. We need to develop ethics and policies appropriate for a species that's both beneficiary and agent of panspermia. We need science capable of detecting and understanding biological exchange between worlds. We need technology that lets us track these exchanges without interfering harmfully with them. And perhaps most importantly, we need humility. The bacteria from 3i Atlas, if they successfully colonized Mars or Earth, accomplished something we haven't yet. They crossed interstellar space. They survived conditions that would kill most of our technology. They adapted to an alien world. These are achievements that deserve respect, even if the organisms involved are microscopic and non-conscious. They represent life's incredible resilience, its ability to persist against overwhelming odds, and its drive to spread and adapt wherever conditions permit. When we eventually drill into Mars's subsurface ice and find bacterial communities thriving there, communities that might trace their ancestry to 3i Atlas or other cometary deliveries, will be meeting distant relatives. Not relatives in the sense of sharing a recent common ancestor, but relatives in the sense of being fellow travellers in the cosmic dispersal of life. We're all bacteria, really, when you think about it. Complex assemblages of bacterial cells working together, carrying genomes assembled from billions of years of genetic exchange, products of a process that extends far beyond any single world. The story of 3i Atlas and its potential bacterial cargo is still being written. The organisms, if they survive, are adapting and evolving right now. Some might succeed spectacularly, founding lineages that persist for millions of years. Most will probably fail, joining the countless other organisms that tried to colonize new worlds and couldn't quite make it work. But succeed or fail, they're part of something larger. The great experiment of life spreading through the galaxy, testing every world, finding every niche, persisting wherever persistence is possible, and we're part of that experiment too. What we do next, how we explore Mars, how we handle the potential for biological exchange, how we think about our place in the cosmos, all of this matters, not just for us, but for the future of life itself. We have the power to either facilitate or hinder the spread of biology through the solar system and eventually beyond. That's a responsibility we didn't ask for and might not be ready for, but it's ours nonetheless. So when you look up at the night sky and think about 3i Atlas somewhere out there, moving away from us back toward the stars, remember that it might have left something behind. Invisible passengers, frozen bacteria beginning the long process of adaptation to worlds that aren't their home. They're out there now, in Martian ice or Earth soil, quietly surviving or quietly dying, writing the next chapter in a story that's been unfolding since life first emerged wherever and whenever that was. And that story includes you. Your microbiome carries bacteria that might have come from comets. Your genes might include sequences inherited from organisms that arrived on Earth via panspermia. You're not just a human, you're an ecosystem, a walking planet, carrying within you the biological legacy of cosmic exchanges stretching back billions of years. The bacteria from 3i Atlas, if they ever here, are just the latest addition to a collection that's been building since Earth first formed. Think about that. Really think about it. Then decide what it means to you and what you're going to do with that understanding. Because that's what this is ultimately about. Not just bacterial dispersal or planetary science, or any of the technical details we've covered. It's about understanding our place in a universe where life spreads, connects, and persists across unimaginable distances and timescales. It's about recognizing that we're part of something much larger than ourselves, something that started long before us and will continue long after us, something that makes every world potentially part of one enormous, interconnected, living system. Welcome to the cosmos. You've always been part of it, now you know.